Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, welcome to the TV learning programs. As usual, I'm teacher Herbert, and I'm going to be taking you through the biology and health science of Senior 2. And the lesson of today, we are going, that is Unit 5, which is concerning about identification of food components. And under this, we are going to go through a practical lesson, which is concerning about testing of starch and reducing sugars. And that lesson is very important in very many classes. It is a cross-cutting lesson that goes to Senior 1, and I think some of you remember you studied it in Senior 1. And I also engage other students in other classes. Take an example in Senior 3, Senior 4, up to Senior 6. It is a lesson that is run in very many classes. So I welcome you to that lesson of practical lesson, and it is of Senior 2, which is food testing or testing some of biological molecules. So let's talk about the lesson, and we are going to go the introduction. Let's go back and talk about the objectives. We have a number of objectives that we are going to go through. These objectives, we have objective number 1 is to state the chemical reagent used to test for starch, testing reducing sugars, non-reducing sugars, and we list the suitable apparatuses. What are the apparatuses, laboratory apparatuses and equipment that are required for chemical testing of a given food sample? So that's what we're going to discuss. You also have to carry out a test for identifying starch, reducing sugars, and non-reducing sugars. But today we are going to do two practical lessons. One is testing starch, another one is testing the reducing sugars. Students, I hope you do remember what are the major components of starch and what are the major components of reducing sugars. Let's go through the introduction. Students, you know that as ourselves, we normally take very many, every day we take, we eat food that contains different components. And these components as food substances or nutrients that helps us to build our body parts. And these nutrients or food substances, we have carbohydrates, we have proteins, we have lipids, we have vitamins, and mineral salts. All of them, they are very important to our body because they are very important to perform the function of a balanced diet. If we eat these food nutrients, you don't suffer from some of the diseases. Take an example like kwashiorkor, take an example like marasmus, take an example, and other diseases that are caused by lack of mineral salts in our diet. So it is very important. Let's go to the second step. This unit is divided into 3 lessons. And we will be dealing only with one of them, which is comprising of testing of carbohydrates, starch, and reducing sugars, as I mentioned it earlier. In this lesson, we have to revise how to test for the presence of starch in food. How can we know that the food contains starch? How can you, because we normally eat very different kinds of food that contains starch. And that's why we have RSB, Rwanda Standard Bureau, that is able to measure and say, this food contains starch, this food contains proteins, this food contains mineral salts. So we are going to test now starch. Can we say the food contains starch? Can we say food contains carbohydrates as an example of reducing sugars? So have you ever studied this unit, student? If you have ever studied that unit, it is very easy for you to understand what I'm telling you. Good. I know that the food we eat every day contains different kinds of different components known as food substances. As I mentioned, we have carbohydrates. These are energy-giving foods. Proteins, body-building foods. Lipids, energy-giving foods. Vitamins, they protect the body against diseases. And we have mineral salts. Examples of mineral salts, you can have like iron, which is responsible for blood clotting. But our today lesson will only focus on carbohydrates. So the lesson of today will be focusing on carbohydrates. And carbohydrates are one of the food substances that you do think that they are categorized into different categories, and we are going to give some respective examples of them. Why some of the carbohydrates are said to be reducing sugars, and why some of them are non-reducing sugars. Let's go through the lesson. The categories of carbohydrates are categorized into 3 classes. Student, let me mention the 3 classes of carbohydrates. We have monosaccharides. Monosaccharides are simple sugars. Simple sugars. Disaccharides, complex sugars. And they are made up of 2 molecules of sugars. They are made up of 2 molecules of simple sugars. We have polysaccharides, and polysaccharides are also complex sugars, and they are made within glucose by condensation process. The process how these polysaccharides are formed by the process of condensation process. So it means that once you get one glucose plus another glucose plus another glucose, you're going to get what you call a polysaccharide. We have examples of these. We have starch, we have glycogen, we have cellulose, we have chitin, we have myelin, and also peptidoglycan. These are examples of polysaccharides. Poly means many. Now, all monosaccharides and disaccharides are reducing sugars, except the sucrose, which is a non-reducing sugars. Student, let me repeat the point. I said all monosaccharides and disaccharides are reducing sugars, except the sucrose, which is a non-reducing sugar. And because they are able to be reduced, the sucrose is enabled to be reduced. That's why we also carry some chemicals in order to break it to be reduced into a monosaccharide. You reduce a non-reducing sugar to a reducing sugar. So for the sucrose, which is a disaccharide, it has to be reduced into a reducing sugar or a monosaccharide. These are the chemical tests that we normally use. We use copper to sulfate in a Bendick solution directly when you heat, and that one is able to reduce a sucrose into a reducing sugar. We have copper 2 oxide. This means that the blue color of the Bendick solution changes to green, to yellow, and finally to orange or red, depending on the amount of the sugars that is present in a food sample. So changing the color from blue to green, to yellow, to orange or red also depends on the amount of glucose or the amount of sugar or glucose that are present in the food sample. When the amount of the glucose or sugar is very high, that one makes the change from one color to another color. Good. What now, the question is, what is food testing? What do you understand by food testing? Why do we have to test the food? We test the food for a number of reasons. We test the number of food to see if the food contains the real nutrients. We test the food to see if the food is not expired. If it is expired, it will be poisonous to our health. It will destroy our body functions. So we have to test the food to see if the food is safe for our health. If the food is not safe for our health, we are going to be poisoned and can lead into death. So this is a definition of food testing. Or the definition is the process that is carried out in laboratory to determine the food components in the food sample. What are the food components that are present in the food sample? Why do we say these are carbohydrates? Student, why do we say these are proteins? So that's why we have to do what you call testing. Another question, student, is testing of food sample. Number one, I will be testing activity one. We have to test what you call starch. I said we're going to test starch. And what indicates that the food contains starch? Student, think about it. Then after think about it, what can we use? What are the chemicals? What are the apparatuses that are going to be used to test the food if the food contains the starch? And here are the materials. And here, these are the procedures. We are going to follow the procedures, and we test our food, whether the food contains starch. Number one chemical one that can just be used to test the presence of starch in a food sample is the iodine solution. Student, I know some of you've never seen an iodine solution. I have an iodine solution. I'm going to show you the iodine solution. The iodine solution is brown in color. So student, if I try to show you the color of the iodine is brown in color. So that is the reagent we used to test the presence of starch in a food sample. So quickly, let me just try to power the iodine solution, and you try to see, this is the iodine solution I'm talking about. You can see and observe. This is the color of iodine solution. So the color of the iodine solution is brown in color, and we use it to test the presence of starch in a food sample. Students, let's go quickly to the procedures. I said it is a brown reagent. Second, we're going to use the test tubes. I know all of you know what are lab apparatuses, lab equipments. These are test tubes we're going to use. We're going to use these test tubes to conduct our experiment of food testing. And we also have to use the droppers. If you don't know the droppers, these are droppers. I'm going to use the droppers. Thirdly, we have a beakers. I have also examples of the beakers. We use these beakers to conduct our experiment, student. I think you are following what I'm doing. And probably if you have some food that contains starch, take an example like Irish potato. Irish potato contains starch. Sweet potato contains starch. Cassava contains starch. All of them, we can use them to test the presence of starch in a food sample. Let me just go to number 2. Now the procedure, way of how you can conduct the experiment. You put 2 milliliters when you have a measuring cylinder of Irish potato, or you can use a prepared starch, extract in a test tube, and add on a few drops of iodine solution and shake and record your observation and draw the conclusion. That's what I'm going to do. I'm going to get a test tube. After getting the test tube, I'm going to prepare a starch. This is a prepared starch. I'm going to use the starch. Then after getting the starch, then I test if there is presence of starch, and then I do what you call a conclusion. I get a test tube, I get a beaker, then I add some starch there. This is a starch. Then I have to add some water. And I'm going to conduct the 2 of them. Then I have to stir. In the process of stirring, I have to stir the experiment. I have to stir, then after stirring, then I will add the iodine solution. I'm stirring, as you see. After stirring, now I will do my experiment. I get a dropper. After getting the dropper, I put there a sample which is almost measuring like 2 milliliters of the solution. 2 milliliters of the solution. Then from there, I add the iodine solution. I add the iodine solution. If I add it, this is the brown color of iodine solution. Then if I add it there, you see, you can observe. The color is changing from this is a blue black color, which indicates the sample contains starch. But if I get another sample, where there is, I add one, I get another test tube, I add a distilled water. Let me use a distilled water. And then I add a distilled water. If I add a distilled water, then I add the iodine solution. In it, we are going to be using it as a control experiment. Then I have to test, I add the iodine solution. You can try to check. The brown color of iodine solution persists. So it means that in the distilled water, there is absence of starch. So you can compare the two. Compare the two. This is, the brown color persists of iodine solution. So meaning that solution, there is absence of starch. But this one is blue-black color, which indicates there is presence of starch. Quickly, let us try to make a deduction, and we make a conclusion. Let's go quickly to our observation. Here you have seen, here there is absence of starch. Here there is presence of starch, blue-black color, which indicates the presence of starch, as you've seen how I did it. Add iodine solution directly to the food sample, and a dark blue color indicates the presence of starch. Note, I think from there, as well we have done it, let us make a note. If the brown stroke yellow color of iodine, the brown color of iodine, solution changes into blue-black. As you've seen it, I've done it, and it is perfect. There is presence of starch in the food sample. If there is no starch, I've seen that I used the control experiment of using a sample of pure water or distilled water, the color of iodine solution persists. And the brown color of iodine solution persists. What is the deduction and conclusion from there? I can mention the conclusion that the observation, changing of the color from brown to blue-black, the conclusion is that the change in color indicates the presence of starch in the food sample, as you can see how we conducted the experiment. And in a starch food sample. Let's go to the experiment number 2, testing of reducing sugars. Testing of reducing sugars. In testing of the reducing sugars, it is very simple. In reducing sugars, we said the material that to be used, we use a solution which is called the Bendix solution. Student, if you don't know Bendix solution is brown, is blue in color. I'm going to show you the Bendix solution. This Bendix solution, I'm going to use it to test the presence of reducing sugars. That is say, we try to make it. This is Bendix solution, which is blue in color. Now I will get a sample which contains reducing sugars. Take an example, like glucose. I know some of you eat biscuits. Biscuit contains glucose. So I'm going to use biscuits. I can use biscuits. I can use prepared starch, prepared glucose, and I say that biscuit contains reducing sugars. I will use a solution of what we call a Bendix solution. Let us go to the materials that are going to be used. One, we are going to use a solution which is called Bendix solution. We have seen that the Bendix solution is a blue reagent. We are going to use the test tubes. And the test tubes, we also use the test tube breakers. That one is a test tube breaker. And the source of heat. Here, you can try to see, this is a heat, a Bunsen burner. This is water boiling. And I'm going to use droppers, a beaker, water, and reducing sugars. I told you, I've used the biscuits. I crush the biscuits, then I'm going to test if the biscuits contains, does the biscuit contains reducing sugars? If they contain reducing sugars, we're going to make a reduction and a conclusion of saying that, sure, we eat biscuits, but biscuits contains what you call reducing sugars. And that's what we're going to go through. Now we go to how I'm going to conduct it. I'll be putting 2 milliliters of reducing sugar, glucose biscuits, and solution in a test tube, and I add 2 milliliters of bendic solution. Then I heat then I shake, then I heat. Then I record the observation and I draw the conclusion. Quickly, let me just do the experiment of how can we test as you see, surely I have something here in front of me. I'll be trying to do the experiment clearly, and everybody can just observe and see if surely this is a crushed biscuit. This is a crushed biscuit. Now I'm going to test, do the biscuit surely contains reducing sugars. Let me try to see if surely it contains reducing sugars. Then after doing it, then I test sure do the biscuit contains reducing sugars. I measure like 2 milliliters. I'm putting it in the sample. Then after from there, you can see this is a sample of it. Then I have to add bendic solution. Then after adding bendic solution, you see the color of the bendic solution. You can see it is blue in color. Now after from there, I have to get another one, and I put it there. I put it in a boiling water. Automatically, I have to make an observation. The color changing. Observation is concerning about color changing. Color changing, color changing, color changing. Okay? It will be blue. Then I also have another test tube where I have to put a distilled water. Then in the distilled water, I also have to add, do the distilled water contains reducing sugars? Then after from there, I have to add bendic solution. Also, I have to add the bendic solution. Bendic solution. You can see the bendic solution is blue in color. Then I also put it in a boiling water. And then I try to make observation. When I'm making the observation, we have to see color changing. Color changing. Color changing is very important. Color changing. Color changing. Color changing, that is the important thing, is to see color changing. Changing from one color to another color. Changing from one color to another color. That one will be even try to show you that the food is containing the presence of reducing sugars. The presence of reducing sugars. Now you can see there's a color of green which is appearing. From green, it will be yellow. Then from yellow to orange. That one will be trying to show you that the food contains reducing sugars. Quickly, if you get the green color, let us be meanwhile seeing how it will just happen. Observation under the color of changing from blue to green to the blue is the color of the bendic solution, and then to green, from green to yellow, then from yellow to orange, then from orange. Automatically, we have already seen the color of green. The green color is being observed. The color of green is being observed from the observation we are doing now. The color is changing from there to the color of green. Then from green, it will change to, as you can see, from green. Then we try to see, to hit and observe clearly. To hit and observe very clearly and see what is happening. Automatically, you can see there is something which is changing automatically. The orange color, which is orange, that one indicates our biscuit contains reducing sugars. But this one, the color of the bendic solution persists. What does it mean? It means that here, there's the presence of reducing sugars. Here, there's absence of reducing sugars. We can make a conclusion. We have done 2 practical lessons. I think you have seen it. And everybody can try to observe from in front of you. We have, number 1 was testing the presence of starch. Turns into blue-black. This one persists, there is absence of starch. And this one changed into orange, which is indicating the presence of reducing sugars. Here, the color of the Bendick solution persists. There is absence of reducing sugars. Quickly, let's go to our lesson. Change of the color from blue to green, to yellow, to orange. I told you changing of the color depends on concentration of the glucose present. We are going to make conclusion and a deduction. This change in color indicates the presence of reducing sugars. We have seen orange, student, we have seen orange color, which indicates the presence of reducing sugars. Here I have something that can summarize each and everything. We have blue. When you put that, there is blue color, which is the Bendick solution. It changes into green, to yellow, and finally to orange. This one also depends on, we call it to be moderate orange, because it depends on concentration of glucose. When the concentration of glucose is very less, as you can see, there is zero concentration, 0.5, the 1.5, then 5 to 2 grams, then 2 grams. As you increase the concentration, the change of the color increases. Quickly, let's go the deduction. The change in color indicates the presence of reducing sugars in the food. And here we have the home assignment. Take your exercise books. I will inform you to take your exercise books and do the assignment. Number 1, give the name of carbohydrate testing which does not require heating. Number 2, describe the color changing during the test for starch. Number 3, what is the color of the Bendick solution? Students, I think that is, you also have to go through that one. That question, we are going to do it in the next lesson, not in this lesson. So student, I am very happy to thank you very much for watching and learning. And I wish you a blessed day and stay safe. Remember to wash your hands with the clean water and soap. May God bless you the whole day. See you next time.